Okay, so let's move on to the pathophysiology of the virus. The virus is transmitted through respiratory droplets. So if you are in contact with in close contact with someone and you got exposed to these droplets, or if these droplets have landed on a surface and you touched that surface and by mistake you touch your mouth or nose, so the virus may get an access to your respiratory tract. It will move down to the alveoli here, and at this alveoli it has the ACE2 receptor, if you remember, and the spike protein will allow the virus to bind to the ACE2 receptor and then to be internalized inside the pneumocyte 2 cells. So once inside the cell, the virus is of plus sense RNA molecule, so that means that, or plus sense polarity, so that means that the virus will be very easily and quickly translated, so that the genome of the virus will be easily translated to make the different proteins of the virus. These proteins will be packed as usual, and the virus will leave our alveoli cells. Obviously, the damage that the virus will leave behind, all of that will recruit immune cells, all of that will stimulate the immune response, and they will produce, these immune cells will produce a group of inflammatory modulators. Without going into details about the names, these inflammatory modulators will cause vasodilation. So vasodilation means that the walls surrounding the blood vessels will be thinner because the blood vessels have been vasodilated. The walls are now thinner. The capillary permeability will increase. The contents and the liquids inside the blood vessels will leak out, and that will create a pressure on the alveoli that will cause the collapse of this alveolar, and that will cause the difficulty in breathing or the apnea. So that is one symptom that many people infected with the coronavirus will feel, which is difficulty in breathing, and that's why we need the ventilation machines and all of that. Also, the damage of the cells of the alveoli will stimulate the coughing, which is also another feature or characteristic for the infection of the coronavirus. And all these modulators, when they go to the hypothalamus, they will increase the body temperature and causing fever, which is the third characteristic of the SARS-CoV-2 infection. Then the virus moves through the blood vessels and capillaries. It will reach the heart. It will cause problems there in the heart, in the kidney, in the liver, so problems in different organs. So, for example, it has been shown that many people infected with the virus, they suffer from kidney failure or liver damage. Moving on to the case fatality ratio. So this is defined as the number of deaths out of the total number of people infected for a given period. So as you can see, there is a wide range of differences regarding the case fatality ratio if you calculate it for different countries. For example, here in Mexico, it has a case fatality ratio about 8.5%. In another country like Turkey, it is as low as 1%, and you got this wide range of case fatality ratio. Regarding Canada, it is around 2.6 or let's say 3%. So this is regarding the case fatality ratio of SARS-CoV-2 between the different countries. But when we compare the case fatality ratio of SARS-CoV-2 against other viral infections, so we'll find some diseases like Ebola, they have a case fatality ratio of 50%, something like MERS and SARS, and as you remember, these are two types of diseases caused by members of the same family of the coronavirus family. So the case fatality ratios of these viruses are really high compared to the SARS-CoV-2. So the case fatality ratio of SARS-CoV-2 is something 3.4 or 1. As we mentioned in the previous slide, it ranges differently in different countries. But the most important thing that it is actually higher than the seasonal flu. So if we want to compare the case fatality ratio of SARS-CoV-2 with seasonal flu, yes, the SARS-CoV-2 has higher case fatality ratio, but still it is lower than many other viral infections. The good news is that we have discovered vaccines, and the efforts for vaccination is actually going well in different countries. And the two vaccines that we are going to show here as examples is the first one is the one from Moderna, Pfizer. So if you remember, this is an RNA virus. It is a plus sense RNA virus, and the most important player in the entry into the host cells is the spike protein, shown here. So what the vaccine developed by Moderna and Pfizer has utilized is the code that codes for this spike protein, and they made a messenger RNA coding for this spike protein. And as you know, messenger RNA or RNA in general, they are not stable molecules. Because of that, they had to package it inside lipid nanoparticle to be stable during injection, transport, and all of that. And because of that, this vaccine needs to be stored at freezing temperature because of the stability of the RNA molecule. So once the person is injected with the vaccine, the vaccine will deliver the code for the spike protein into the host cells. The host cell will translate this protein, or actually translate the messenger RNA into the spike protein. So all of a sudden, our immune system will find many proteins which are non-self, they are foreign inside the body, so the immune system will respond and produce the antibodies like that. So once you develop antibodies for the spike protein, now you are ready for the actual contact with the SARS-CoV-2 virus. So if happened and you came in contact with the virus, now you have antibodies ready in your blood, and they are waiting for the virus in order to bind and inactivate it. So you may be wondering what will happen for the messenger RNA. Once the messenger RNA is translated, then it got degraded, and that's a normal thing for the messenger RNA. They are degraded, so the messenger RNA will no longer be there after delivering its role, which is the production of the spike protein. The second vaccine we are going to discuss is the one developed by Oxford or AstraZeneca. So this vaccine uses the technology of adenovirus. So we have edited adenovirus, and this is a virus that can enter our host cells, our host human cells, but it cannot replicate inside them. So what the scientists have done, they have taken the DNA sequence or the gene sequence responsible for the production of the spike protein, and they have put this DNA sequence inside the adenovirus. So now once you are injected with this edited adenovirus vaccine, what will happen? The DNA coding for the spike protein will be released from the adenovirus inside the host cell, and your body is going to synthesize many proteins that corresponds to the spike protein of the virus. and because of that, and all of a sudden this foreign protein starts to accumulate inside your body, so your immune cells will respond producing the antibodies specific for the spike protein. And if it happened after that you came in contact with the real coronavirus, now you have the antibodies in your blood waiting to take action. So I'm going to finish with this slide, reminding ourselves what can we do now, or actually what we should continue doing till we all get vaccinated, and hopefully we will end this pandemic as soon as possible. So the social distancing and wearing the face mask and getting vaccinated if possible, and also using the alcohol-based washes. And now you know that all this alcohol-based washes and the mechanism they work with, so all of these measures will help us to overcome this viral infection, and hopefully as soon as possible. Please stay safe and please stay healthy, and thank you.